Hi everybody, the topic for today's lecture is DNA replication. So let's get going on that. So what we see here is a sperm fertilizing an egg. We all obviously know this story. What you may not remember is that each sperm, for a human being anyways, contains 23 chromosomes, right? And the egg contains 23 chromosomes. And so once this turns into the zygote, these get fertilized and combined together, you have 46 chromosomes total. And the blending of these genes on these chromosomes produces your own traits. And so you're sort of this mix between your mom and your dad. Okay, but how does that one zygote, that one first cell that's formed, turn into a person? And so we start out as one single cell, and then we end up as trillions of cells, right? Okay, trillions of cells, and all of these cells are doing different things, right? Cells in your skin are doing different things than the neurons inside this baby's brain. So how do we get to that point? And so you guys know by now that this one cell is going to divide. We're going to do mitosis. Whoops, wrong picture. Right here. So at first we're going to divide in two. Those two divide into 4, then 8, and then 16, and so forth. And so the first bit of cells, these are called zygote, and then it's going to turn into the morula, the blastula, gastrula, and then eventually we're going to get the embryo that's going to start to eventually look more like a baby, and that's going to turn into a fetus. So when this division goes on, so when this mitotic division happens, we need to make sure that this cell and this cell, we need to make sure that these two cells have the exact same DNA. And so then these 4 cells have the exact same DNA. After that, these cells are going to start to communicate. We're going to have differential gene expression. So eventually, these cells are going to start doing different things. But for a while they're identical. They have the same DNA. Nothing's really turned on or off as far as the genes go. And so these are just the most basic stem cells here. Okay, so how do we make sure that every cell in our body has the same DNA? All right, so that happens right in here during S phase. So this is when we identically duplicate every single chromosome we have. So you're going to make an identical copy of those 46. Okay, so let's go over real quick. So when do we replicate our DNA? We do it any time a cell is dividing, okay? Okay, so this is pretty much going on all the time in any of your growing cells, especially things like skin that gets replaced a lot. Or your intestinal lining is going to get replaced a lot. So you're going to have a lot of DNA replication going on in those cells. Something like a brain cell, you're not really doing DNA replication because your brain cells don't divide. You guys are already at the point basically where the number of brain cells you have is constant. All right, just downhill from here. Sorry to inform you. Take care of those brain cells. But anyhow, this is going on in the nucleus, okay? Again, in the nucleus of all the cells that are dividing. And why? To make sure, or to ensure each new cell has a complete set of chromosomes. Okay, so each cell is going to get the entire recipe book of genes. It's not necessarily going to use the entire recipe book. It's only going to take what it needs, but it's going to contain all the DNA, all the same genes, all the same exact sequence is found in any of your cells. This is why if you leave a little hair follicle at a crime scene, they can trace it back to you because that contains all the DNA, not just the DNA that codes for hair. That would be identical for many people, but it's going to have everything. All right, so we completely have to copy the DNA. So let's review a little bit about how DNA is, okay? So what you have here, here's our double helix, right? That's the shape, is a double helix. It's kind of like a twisted ladder. All right, and let's take a look. So the double helix, or the DNA, is a polymer. It's a macromolecule. It's really, really big. You're just looking at a little section of it here. And so let's name all these pieces. So this right here is deoxyribose. Remember, that's a sugar. Okay, here's a phosphate. Okay, up here, that's a nitrogen base. Okay, so that's the thymine nitrogen base there. And all along here, the backbone, all these solid bonds right here, these are all covalent. Because what really matters in DNA is the sequence of these A's, T's, G's, and C's. So that sequence is going to be held together with very strong covalent bonds, because we don't want that to shift around. When we start to shift around these A's, T's, G's, and C's, that's when you get mutations, and that's not always a good thing. So we make sure that these are strongly bonded together. Okay, now in between the two sides, these are hydrogen bonds. So this is another case where structure and function are very closely related. There's a reason why hydrogen bonds are good here, and that's because DNA needs to unzip to copy itself. So as we replicate, these are going to come apart quite easily, because remember, hydrogen bonds, the same thing that holds two water molecules together, these are weak. They're easily separated. Okay, water can fairly easily evaporate, for example, so those weak bonds can break. All right, the other thing I had to tell you guys was DNA is what we call anti-parallel. It's kind of like streets that have a lane going this way and a lane going this way. You can see that these phosphates, all this stuff is upside down. Okay, so the nucleotides actually run the opposite direction. That's one thing I left out. You guys should review what a nucleotide is. So each set of nitrogen-based sugar and phosphate is a nucleotide. Okay, cool. So let's go back to this thing, anti-parallel. What does that mean? And you'll see these little marks, 5' and 3', we call it. And that refers to this sugar right here. So organic chemists basically assume that you understand that there's carbons in here. So this is a 5-carbon sugar. So you don't have to draw the carbons and hydrogens in. We just assume you know that they're there. And then when we want to talk about certain carbons, we just go around in a circle clockwise from the oxygen. So we say that's the 1 carbon, this is the 2 carbon, the 3 carbon, 4, and there's one in this corner too, that's the 5th carbon. Okay, so you can see the 5th carbon kind of sticks up here. On this side though, okay, we always count clockwise from the oxygen. So that's 1, 2, 3. The 3rd carbon is at the edge here. So that's why this is the 3' end, this is the 5'. So when you come down here, this is the 5 carbon. So 1, 2, 3, 4, 5, okay? So that's why we have to name these sides, because they're slightly different and they go opposite directions. Now that you might not know why that means something right now, but it's going to mean something later when we replicate this DNA. All right, so just hang on to that information. There's a 3' end and a 5' end, okay? Let's move on. All right, so now you see this 3', 5' stuff again, okay? And so the way DNA replication happens is basically the DNA unzips, we put new nucleotides on one side, new nucleotides on the other side, and these should match each other exactly. So imagine you have G, A, T right here, GAT, right? So over here would be the match to that. G always matches with C, A always matches with T, T would be A there, okay? So this would be the complementary strand right here, okay? These two pieces used to be together. All right, so when we put new nucleotides on, then there would be an A here, a T here, a C here, okay? And the way the enzymes that do this, so there's enzymes that actually come in and put in the new nucleotides, what's gonna happen is they only work from 5 to 3, okay? So this is something to note. All DNA replication, oops, I can't write over there, so it's DNA replication enzymes work in the 5' to 3' direction. Okay, and that's in reference to the new strand you see here. So don't look at the old strand, but if there's a 3' end here, that's going to match with the 5' end here. All right, so 5 to 3 over here, we're going to start to put on those nucleotides, okay? And so enzymes are going to form these hydrogen bonds. And let's see what's going to happen here. Now, this looks a little different, right? These are called Okazaki fragments, and those form basically because we have to wait for this DNA to unwind a little bit at a time. And so as it unwinds, we can make the match, okay? Wait for it to unwind, make that next bit, wait, make the next bit, okay? So we can't do it continuous like this because remember, the enzymes only work in the 5 to 3 direction. So we actually have to start from over here, and if it's wound up, we might have to wait a little bit. Okay, so this is kind of made in these little tiny pieces. All right, but nonetheless, we're going to synthesize this piece as well. And so C is going to match with G, so all these enzymes are going to put down the matches, okay? A and T, and then T is going to come here. So you see at the end we have two identical sequences here. And so if we go down the whole entire length of the chromosome, we're going to have identical copies by the end of it. All right. So since this one can go smoothly 5 to 3 all the way across, that's continuous. And we call that the leading strand because it's just going to, it doesn't have to wait for anything. It just continues on, and as it unwinds, it just goes for it. This one, it's not one continuous piece. It's going to be in fragments. And so since this piece has to kind of wait for it to unwind a bit so it can, that enzyme can get in there and lay down some track, it's called the lagging strand. All right, and where this separates, that's called the DNA replication fork. Okay, so let's get into what these enzymes are. That's what's next. All right, so here's the same piece again, and it might be good to grab a few different colors at this point, just to kind of help your notes make a little bit more sense. But remember, these are going to be hydrogen bonds holding these two sides together, and there's an enzyme called helicase. Okay, so H stands for helicase. Okay, and that's going to unzip the DNA. Okay. Okay, and there's going to be these proteins called single-stranded binding proteins, and they're going to make sure that they hold the DNA open and stable, okay? Okay, so they're just going to be pulling this DNA apart a bit,